Okay, I think we can go ahead and get started. Hello, everyone. Welcome to another session of our Evolution Seminar series. Today, we have a special Early Career Award seminar. This award is made possible by contributions from the College of Agricultural and Life Sciences, the College of Letters and Sciences, the Nelson Institute for Environmental Studies, and the Graduate School of UW-Madison. Really excited to introduce our speaker today. Marco González-Santoro is joining us from the Department of Biological Sciences at the University of Pittsburgh. He's going to share some fascinating research on sexual selection and speciation. So let's all give Marco a warm welcome.
Right. Thank you so much for the introduction, for the invitation. It has been great to meet some of you all this morning. I'm really excited to continue meeting some of you. And today I'm going to share my progress so far in my PhD, which I so far have titled The Role of Social Learning in Speciation by Sexual Selection. And I would like to start pointing out the reason why I'm interested in speciation and why I think it's important. And it's because speciation is the mechanism behind the generation of all of the biodiversity that we know on Earth, right? Which is pretty fascinating. And natural history museums make a good job on giving us a glance of what it looks like. And so far, as today, we know that there is 9 million of described species, which is fascinating. And even more fascinating than that is that the 9 million of estimated species today are estimated to be only 1% of all of the biodiversity that has existed throughout the history of Earth. So speciation is an incredibly powerful and fascinating process that has interested biologists for many years. So it is not surprising that the very foundational work of evolutionary biology, which started with Darwin, was focused on answering these very same questions. How do this biodiversity arise? How do species exist? So after 150 years of research, we have been able to simplify this very complex process in key steps that are needed for the speciation to be completed. First, we start with divergence in traits. This is not particularly rare. This happens in almost all populations. There is some standing genetic variation in populations. The key for speciation is whenever those divergent traits evolve reproductive isolation between them. So the main question in speciation has been, how does this occur? Historically, this separation and this reproductive isolation between the divergent phenotypes has been explained under a geographic context, and that is because geographic barriers are a very good mechanism to separate the divergent traits. So we have this very classic example from Humboldt in his expedition, 19th century in Ecuador and South America. This is the Chimborazo, a famous mountain in Ecuador. And something that he recognized is that divergent traits not only could be separated at the different sides of the mountains, but if you cannot really see, but all of the white part here with letters are species. So he recognized that the altitudinal gradients in the mountain could be a climatic gradient for species to be separated. So they could be adapted to different climates and eventually have this physical separation. And a very good example as a geographic barrier are rivers. So rivers can also mean geographic barriers for species. I like this example of these birds in the Amazon basin in Brazil, because you would think that birds can just fly between the different places of the rivers, but apparently rivers are making their job of having these different species separated and specialized in isolation. So the geographic context works, but it's incomplete. And we can see this by the work of these also two foundational authors in evolutionary biology, Meyer and Dobzhansky. They were part of the modern synthesis that brought Darwin's ideas and Mendelian and population genetics together. And 20 years apart between the two of them, they were thinking about the same ideas. In this case, we have an initial population, as we saw, and they envisioned how this initial population could diverge into two different species. What they both recognized is that during the process of divergence, in multiple times, the divergent phenotypes were going to be together, either in the moment of divergence or even after geographic isolation in secondary contact. And Meyer even recognized that secondary contact could merge again the divergent phenotypes. So this is a problem for speciation because they can separate, they come together and the species disappear. So the geographic context of speciation has shifted a little bit from understanding how the barriers are important to understanding how do reproductive barriers arise in the absence of geographic isolation. And to answer this question, sexual selection offers a simple but elegant solution to the problem. And this is because sexual selection facilitates the divergence of mating traits, in this case, coloration, for example, the color of the frogs there, but also sexual selection facilitates the divergence in preferences for such traits, which eventually can lead to a specific type of reproductive isolation, which is behavioral reproductive isolation. I like to make emphasis in this one, that is only a particular type of reproductive isolation, because plant people always gets mad at me when I say like this is how speciation works. Plants are crazy, they do all kinds of stuff. So this works pretty well for animals. And it's only one type of reproductive isolation. But coming back to this process, the divergence in traits and preferences is usually, and it's very intuitive to think about female choice. In this case, we have females with different coloration, so the orange and the blue females, and they develop divergent preferences. In this case, illustrated by the color hearts. So essentially, a female that is orange that develops a preference for orange males is going to ignore the blue males, and then gene flow cannot happen between the divergent phenotypes, a pretty easy way to have behavioral reproductive isolation. A second and less studied phenomenon of this is male aggression. So in this case, male aggression can happen directly for the access to a female, or for example, for territories. So males that have territories are going to be more successful because they have resources, and females are going to arrive. And the idea here, illustrated by the gloves, the colored gloves, is that males also biases their aggression towards individuals that look like themselves. So in this case, the orange males only fight orange males, the blue males only fight or blue males, and this is important because this allows the coexistence between different divergent phenotypes. So male competition allows for the coexistence of different morphs, the divergence in traits, that is the very first step, and female choice prevents gene flow between the divergent phenotypes. So we have two ingredients perfect for reproductive isolation. The key question in here that is important up to this day is how can mating traits and preference evolve together. And this is hard to explain because you need to imagine that traits, such a coloration, have a genetic basis of multiple genes of pigments and melanin and structural colors, and preferences, which is how animals perceive coloration, is completely different from the genes of the mating traits. How can they evolve together has been an evolutionary problem for many years. We can simplify this instead of thinking of multiple genes of traits and preferences as two genes. Imagine one gene of major effect that controls coloration and one other gene of major effect that controls preference. We have two loci. And the problem here illustrated by recombination in sexual animals, sexual organisms. Imagine that the color gene is in this part of the chromosome in the head here, but the preference gene is here. Recombination happens, a common process in meiosis. And then the germ cells, this can be sperm or eggs. Some individuals are going to have the color gene of one, but the preference of the other. So this is going to break apart the association between trait and preference, which is a problem. And this can be illustrated here with these butterflies. These are two species that they hybridize. They have F1 heterozygotes individuals. They continue to hybridize. And some individuals eventually will have the gene of one, for example, being red, but the preference of the other, preferring the white individuals. So color and preferences are broken down easily by recombination and gene flow. So at this point, I imagine that you can be imagining of a solution. We have the two loci problem. What if we have one loci instead of two? What if one gene controls both coloration and preference? That would solve everything. And this is exactly what these authors, or not exactly, but something very close to that solution. So in this study, they found that in the chromosome 18, the gene that controls coloration, in this case, this red coloration, was very close in physical proximity from the preference gene. So recombination couldn't break this association very easily. And what they found is that those genes were integrated from one species to the other in the eastern cordillera of Colombia. And so they share the coloration, this red, and they also share the preference for the other one. So this is a solution to have the two loci problem to become one loci problem. But still, it was a lucky chance that they found these two genes together. They don't really know what this gene of the preference does. It might be a lot of stuff happening either in the eye or the brain or somewhere in the middle. So what other solution can we have for this? I'm going to explain to you one in my presentation. But before the mechanism, I'm going to introduce you the model system that I use, of agapomilio, the strawberry poison dart frog in English. The local people call them ranita flecha roja y azul, which means a red and blue frog. And some people call them blue jeans, because it looks like they're holding a pair of blue jeans in their red body. A very charismatic animal. We have here the distribution. So we have them in Nicaragua, then they go down to Costa Rica, eventually to Panama. And what I'm showing you here is the distribution in Nicaragua, all red and blue frogs. We go to Costa Rica, red and blue frogs. It's the blue jeans frog. The reason that I am interested in this species is whenever we go down here to Panama, to this archipelago, whenever we arrive, the red and blue frog turns into this different phenotypes of different coloration, so dramatic variation in their colors. All of this, the same species. My boss did the experiment of crossing all of them together. The babies are okay, the babies can have more babies, no type of barrier between them. An amazing system because we have the first ingredient for speciation, which is divergence in traits, probably facilitated by the separation of the islands. So we have the first ingredient for speciation. And the second reason that I like this species is that they have a specific type of parental care. This is the mom. What the mom does is to carry, she picks the tadpoles, she puts them on her back, and then she goes to the forest looking for these plants called bromeliads. It's like these tanks of water that accumulate water inside of their leaves. And she leaves the tadpoles inside. And what she does eventually is that she lets the tadpole on the water, and she's going to come back regularly to the plant to release unfertilized eggs. So a very specific type of parental care in a frog. It's fascinating. And what is interesting of this is that the feeding of the tadpole is triggered by this. This is called the begging behavior. So it's essentially like the tadpole, this kind of dance is begging for food. And this triggers the mom to release an unfertilized eggs. This is interesting because some researchers, meaning my boss and a previous student, hypothesized that this could be a moment in which the tadpole and the adult interact together, and then the tadpole could imprint on the coloration of the mom and take a sexual decision later in life. How it works is that a tadpole that comes from a red mom is going to imprint on the coloration of her mom, the red color, and later in life, that individual is going to develop either female choice or male aggressive biases towards individuals that look like their mom. To prove that this was true, they did a foster experiment. So the tadpole that was red was raised not by the biological red mom, but by a foster mom of a different color. Later in life, what they found is that the individuals developed female biases and male aggressive biases for the foster color and not the biological mom. So a fascinating mechanism for a frog, for an amphibian that we think they don't do much. So this was fascinating to find, but this is not really new. We know 50 years ago that this would happen on birds. The classic experiment, one species of finch being raised by another one, and later in life, the two species were courting the wrong species. And 300 years ago, this is a painting that I found in one of Pittsburgh's museums. It's called The Lady with a Bird Organ. And the bird organ is just like a speaker of the time. where they could display music, and they have a little bird in a cage. What she's doing is teaching the bird, sing the song that she likes, so the bird can repeat it all day. So birds have shown learning for sexual traits, even though if it's something artificial for a very long time, but until now, we are connecting this as an important mechanism for speciation. So we have that one way for traits and preferences to evolve together is imprinting. So as any good science, this generated more questions than answers. So questions that I'm approaching for my PhD. The first one, I want to come back to the species. I show you that Costa Rica is red and blue frogs, Panama, dramatic color variation. So the question here is almost obvious. What happened here that didn't happen on the rest of the species range? Something interesting would have happened. If we see the phylogeny of these frogs, we have the Panama clade called the Bocas del Toro lineage, which is the name of the archipelago, where we see all of the variation. And we see as outgroups the Costa Rica lineages, all of them red and blue frogs. If we think that learning is facilitating the divergence of coloration through the effect of male aggressive biases and female mate choice, we could imagine, and this is the populations where imprinting was proved in the original paper. So we can imagine that maybe one hypothesis is that both female mate and male aggressive biases evolved only in the Bocas del Toro lineage, which didn't evolve in the Costa Rica lineage, and that could explain why there is variation here and not there. A second hypothesis is that female mate choice is in all of the species, but only male aggressive biases evolved in Bocas del Toro. If you remember, male aggressive biases were the important ones for having variation in the species, because they allow coexistence between the different phenotypes. So maybe that's a second option. Or a third option is that both exist in the species, and it only evolved to be stronger in Bocas del Toro and not Costa Rica. So one question that I have for my PhD is how have learning biases evolved? And the second question, ignoring a little bit Costa Rica and focusing in Panama, you see all of this variation of coloration, and almost in each island there is a different color. And those are islands where all of the frogs look alike. They have low variation in their phenotypes, but there are some populations here in red shown there where there is multiple colors coexisting together. And sometimes it's almost a continuum from the red to a blue and brown, purple of kind of coloration. And this becomes complicated because if we look at the natural history of the frogs, we know that they imprint on the coloration of their mom, but they continue to interact with individuals around them. So males are aggressive, they defend territories, and they're going to go into physical combat, this sumo-like combat that they have. So they interact very closely with other males. Females usually court for days to males. So they will stay close by to a male that they are interested in, stay there for some days until they eventually go into physical contact and they court, and then eventually they release their eggs and everything, but they are also in close contact with other individuals. So you can imagine an individual that imprints in the coloration of their mom a red, but eventually only finds individuals of other coloration. Do they take the decision of mating with whatever is around, continue learning that that's the phenotype around, or they lose the opportunity to mate? So this complicates things a little bit. So I wanted to know from my PhD if imprinting was the only moment of learning, or if this type of sexual learning continued to occur for other individuals at different life stages. So for today, I'm going to show you progress in these two chapters that I have done for my PhD, the evolutionary patterns of learning and learning at different developmental times. For the first question, how have learning biases evolved? Remember that we have these two behaviors important for speciation, female bias, male aggressive biases, and to understand the evolution of this, of course, we need a phylogeny. So we have the phylogeny of the frogs. So here's the common ancestor. These are the frogs of Costa Rica and Nicaragua. You see red and blue frogs. And then we have the Panama clade ancestor with populations that are either one color monomorphic or polymorphic with different colors. Here highlighted with the letter P. And then we have to map these two behaviors in the phylogeny and observe their evolution. I'm going to start with the male aggressive biases first. So as I was telling you, they are aggressive. They have this escalated aggressive behavior. They're going to call from a territory to advertise that that's their territory. Whenever there is an intruder inside their territory, they approach them. And if the intruder doesn't leave, they escalate the behavior to this physical combat. So what I did was to play the intruder myself and act as an intruder of their territories. I did this by having these 3D printed models, essentially like a plastic frog that I painted with the coloration of the population. So this is like an example to see what they look like on our eyes. Yeah, pretty close, but it's also important to remember that what we see is not what the frogs see. So this is usually the absorbance of our cones. In this one, how our cones are absorbing the wavelengths. And our peaks of absorbance is different from the ones in the frogs. And that we know from a physiological experiment in 2006. So we know that they are looking different. So what I do, that we can do now with that information, is that I take all of my 3D printed frogs with different colorations, different shades of green, different shades of red. And I use a spectrometer, a machine that you throw a beam of light and whatever it reflects back is the coloration that something is reflecting. So it will tell you what color is the frog. And then I can use this information and I run it through a model that has the information of the eyes of the frogs. So we have information for this species, but we have information for some birds that they have 4 cones. We have information for crabs, for some snakes. So if you have any questions, we can discuss about it later. It's a fantastic tool. But then eventually we can have this kind of peaks in which we have wavelength. So from blue to red, and then the percentage of reflectance in the machine. And we can compare how close they are to different frogs by having this chromatic distance is just the peak of where the reflectance is showing. So the purple one is probably like a green frog around 550, and the yellow one is probably a red frog. And or we can compare the chromatic distance, which is how bright is the 3D printed model by the area under the curve. And we can eventually compare how 2 frogs look alike to do 2 things. First, to make sure that the models do resemble themselves, to resemble the coloration of the population that we are trying to test, but also to test how are they perceiving different colorations. So something that, for example, was a surprise for me is that the green is most closely related to the red than to the blue in the eyes of the frogs. Because I always thought that the red was the most different of everyone. Apparently, it's not. But anyway, we can do this kind of stuff. And once we confirm that information, we can start doing the experiments. So for monomorphic populations, where there is only one color, I approach territorial males 30 times and 30 times, one with the coloration of the own population and a different population. And in the polymorphic ones, I used colorations that exist in the population, and I just show them the different colors that exist. How the experiment looks like. So I listen to the frogs that are calling and telling us that they are holding territories. And I have like a speaker with my 3D printed frog inside a fake lock, and I start broadcasting the call. So this is a male that was calling. I'm broadcasting the call right now. So they are super aggressive. This is 10 seconds in the experiment. They immediately look for the intruder. Then it arrives to my log. So I was broadcasting the call in here. I have my 3D printed frog there, and I have a motor down the frog to move it around. One of these like toy cars, like the wheels do like this. I just put it down so they can move like that. And then this is what an aggressive response looks like, either for the same color or a different color. They have this sumo fighting, like they try to embrace each other. Yeah, it doesn't look too aggressive, but essentially they embrace each other. And whenever they are like this, they start to call. So imagine like your head is like bouncing against something. I think they get dizzy at least, but that's the fight. So I quantified the number of attacks that they had for my 3D printed frogs with different colorations. And for the female mate choice, I had to do something different. I think the females are more intelligent than the males. The females were not tricked by my 3D printed frogs. They ignored them. So I decided to change the experiment. Instead of that, I used 2 live males. So I put the female inside like a box with lights that resemble the coloration of the light, the coloration of the forest. So I have to put like some blue filters there because the red light gets lost in the canopy and this blue light arrives to a forest. So I have the lights and I have these males that are alive inside like a transparent dome. And I draw an interaction zone. So essentially I sit down for my students for an hour to look what the female does, to who is she interested to interact with. So it's a very fun thing watching a frog for an hour. And eventually I just do simple math to calculate the proportion of either female mate choice or male aggressive biases. And the reason to do this is to have a measure from 0 to 1. So 0 would mean a strict preference for a different morph. 1 would mean a strict preference for their own morph, as we expect from imprinting, or 0.5 means no preference. This value can eventually be put into a color scale. I don't know why that looks like that, but anyway. So we can have a scale from 0 to 1, 0 meaning a different morph, 1 meaning their own morph, and we can plot this in the phylogeny. So we can see, for example, if those populations show a preference for other morphs, this for another morph, we can track the evolution and say how likely the common ancestor was to have their own biases. Cool. So these are the results that I have so far. So again, the scale is there. I'm going to start with the male aggressive bias. So again, we have Panama clade in those 5 populations there. These are only monomorphic populations. We have the Costa Rica there. And what we see just by the colors is that aggressive biases probably existed in the common ancestor. You see this population here, it's kind of orange to yellow. So probably they are all aggressive, as we were not expecting, but definitely the male aggressive biases evolved to be stronger in the Panama clade. As you can see, almost all of the populations had a strong bias for their own morph. And when we see in the for female mate choice, we see that this is pretty green, this is pretty close to 0.5. So we can say that there was no female choice in the common ancestor, but it evolved to be a little bit stronger, or it evolved to exist in the Panama clade ancestor. This is exactly what we were expecting. So this is nice. So it means that female mate choice and male aggressive biases can explain the color variation that we observe, but things change a little bit whenever I include the polymorphic populations. So remember, these are populations that multiple colors coexist. And then these were the predictions that we had. If we focus on the polymorphic populations, this is what we found for the monomorphic ones. But if you focus on the polymorphic populations, you see that these 2 behaviors decrease. All of them go to the blue and green hues. So both female mate choice and male aggressive biases are lost in polymorphic populations. So this was not something that I was expecting, especially because polymorphic populations have the potential for divergence, but they are disappearing. So something interesting is happening in the polymorphic populations, but something interesting too is that these 2 behaviors are evolving together. You see that the pattern almost matches the 2 trees, and this is interesting because these are 2 completely different behaviors in different sexes, controlled by different neural circuits, different genes. So it's pretty fascinating that learning may drive these 2 behaviors together. And I just confirmed that doing like a simple correlation test, it was significant. Whenever I correct for the phylogeny, it stops being significant, but pretty high correlation, 0.5. So it seems that learning is a way in which female mate biases and male aggressive biases can evolve together, which we know both are needed for speciation. We know that imprinting may have evolved to be stronger in Panama, potentially driving color divergence, but this depends on the social environment of the frogs, either if there is monomorphic or polymorphisms. So that leads to my second chapter, especially the polymorphic populations, in which I wanted to know how learning is happening at different developmental types. So for this one, I brought frogs from Panama to Pittsburgh, which was a whole operation, but we have the frogs. And I have breeding pairs of different populations. We have the blue frogs and the red frogs, and we allow them to have babies. So what I do is that I take the babies and I let the babies, the tadpoles, imprint on the coloration of their mom. That means their own coloration. And once they metamorphosize, I start changing their social environment. So I put some individuals in terrariums with individuals that have their own coloration or a different coloration. So some individuals will see their mom and never again will see the same color, at least in the juvenile experience, which is 10 months. And I do the same for the other phenotype. And then after they reach sexual maturity, after 10 months, I do a first behavioral assay, which I'm going to explain in a second, but it's the same, the one of the cage, because now it's in Pittsburgh. So I do both females and males are focal individuals in the interaction box that I had. And after the first behavioral assay, I switch the social environment of the frogs. So frogs that had experienced their own phenotype at the social experience, now they're going to experience 3 months with a different phenotype. And then I complete a second behavioral assay, a super large experiment that has been going on for years and is continuing. But that's kind of the stuff that we have in the lab, a bunch of terrariums with different social environments. And as I was mentioning, the experiment is as simple as for the females in the field, just that in this case, I am also testing focal males and the interactions with different phenotypes. So to explain the results so far, in the x-axis, we have the social environment. So either they socialized with a different color or their own color. And we have the same 0 to 1 proportion, 0 meaning a strong preference for a different color, 1 meaning a strong preference for their own color. So if we were to follow, for example, this part of the experimental design, those are individuals that imprinted on their own coloration, but then for 10 months have only been with a different phenotype. So we would expect both females and males to have a very low preference for their own color if they continue to learn in the juvenile experience. What we have found so far is that yes, this is exactly what I found. So pros continue to learn, so they don't prefer their coloration of their mom anymore, but whatever they more recently experienced. So this was nice to find. And then if we focus on the other experimental design path, these are individuals that imprinted on their own coloration and have never seen anything else again, the same coloration throughout their lives. What I found is that it was true for females and not for males. This was interesting because these males have never seen that color again. So either they suck at learning, probably that's one, but another one is that I'm putting a male. So imagine it's terrariums with these plastic cups that resemble the plants where they put their eggs. It's very likely because the terrariums have a female and a male, and then I put a juvenile there. It's likely that the males that are in the terrariums already have territories inside the terrarium. So the juvenile might as well be scared. So it's learning, but the opposite way, because they are receiving a lot of attacks. That's just one idea because there's no way to explain that. But anyway, that's the data so far. And when we move to the second part of their developmental time, these are frogs that are already adults, the males already call, the females can already mate, and I switch the social environment. So if we focus on this one, so these are individuals that experience a different phenotype during their juvenile stage, and now they experience 3 months with their own color. So we would expect this to switch to this if they continue to learn. What I found is that it was true again for females, not for males. Females are learning super fast and learning about their social environment. Males continue to interact with whatever was happening in the juvenile stage, which was interesting. And if we follow the other one, we would expect the same thing, the proportions of preference to be switched down now because their more recent experience was with their same phenotype. What we found, true for females, not for males. So this is the data that we have so far, summarizing a little bit here. We found that they continue to learn from individuals, not their mom, just individuals in the population. But females are doing what we expect, males do not. My hypothesis is that females are the ones with the parental care. So they need to carry the tadpoles and leave them to the trees, and then you have to come back all the time to feed their tadpoles. So they may have a very good spatial learning, and social learning might as well just be like a part of learning. So if they are good at learning in one context, they may be better at a different context. And males that hold a territory and don't move much maybe don't learn as well. So that's something to explore. So we see that social learning continues to occur at different life stages by non-maternal tutors, and it may differ by sex, and this data that continues to be taken every week until the day I graduate, because it's so long. But anyway, summarizing, we found that this little red and blue frog has diverged has gone through a dramatic color divergence. So all of these are colorations that are found in a population. So it's pretty striking that a red and blue frog can turn into this in very recent times. But, and that it may trigger by the special parental care in which they learn from the coloration around, but that it will depend on their social environment. As a general conclusion, I would say that some social learning environments may trigger speciation rapidly, while other ones do not permit this. Just as future steps and current work that we are doing, one is understanding how do animals perceive coloration, of course. We have these models based on their absorbance of their cones, but we want to do something more behaviorally while trying doing some stuff, so we can discuss more about it later. This is something that is going on in the lab. Also, sexual selection can facilitate divergence in coloration, but these are poison frogs, so they need their colors to survive. So what happens when a coloration that is not as good in the environment is maintained by sexual selection? The blue frogs is essentially a cryptic frog. Are they changing their behavior with their coloration or not? It's something that I am working on it right now, going to Panama soon in the summer. Which is the neural basis of these two behaviors and why are they evolving so easily together is something that I'm approaching too. And finally, we are collaborating with these theoretical models of speciation to include learning as a variable important for theoretical models of speciation. After that, I just want to thank all of the people. There's a lot of time in the field. It's been 1 year or 2 already on the field. So a lot of people that helps me, a lot of Colombian assistants that have gone there, my lab for taking a lot of data, the undergrads, Pittsburgh that pays for my PhD. This is a NSF project, and I go all the time to Smithsonian, so this is nice. And the Wisconsin evolution, this is a Department of Evolution Genetics for the invitation here. And with that, I will receive any questions that you have. Yeah.
The imprinting and the divergence of color. So the fact that you only see imprinting in certain populations suggests to me that there might be selective pressures in favor of imprinting in one population, but not the other. So one possible explanation for this is that if there's some fitness cost to the hybrids apart from sexual selection, that the imprinting was a mechanism for secondary reinforcement to prevent the hybridization. So in other words, what do you think is primary? Do you think there was selective pressure for for imprinting that led to divergence, where you first got the divergence in phenotype and then the imprinting through secondary reinforcement evolved to prevent
Yeah.
formation.
Yeah, that's a good question. So we have done some attempts to understand hybrid costs. So one of them were crossing the frogs, which it didn't seem to have any cost. There was another piece of evidence in which, for example, in the population that is red, blue, and all kinds of intermediates, what we did was to put plasticine models that we painted. We put them on the forest, waiting for predators to attack them. And we should expect maybe the brown ones, purple ones, intermediates to be attacked more than the red or the blue. It didn't seem to have an effect. So it seems that maybe it's not against hybridization itself, how imprinting evolved. One idea is that the adults can predate on the tadpoles if they are not their babies. So an individual will arrive, and if it's not, so an individual will arrive to a tank, and if the tadpole begs to the wrong mom, essentially is going to get predated. So maybe they evolved the ability to imprint to avoid whatever was not their mom or not likely their mom. That's one idea. It was based on some natural history observations that we saw predation of non-conspecific tadpoles. So it could be a reason why tadpoles are recognizing so fast, because it's either recognizing food or badly recognizing something that is going to predate them. It's a work in progress. Yeah, it's
Selection in favor of imprinting all populations. Don't all of them have maternal care and so on.
sorry, can you repeat that?
Well, if that were the case, you would have selection in favor of imprinting in all the populations then, right?
Yeah. Oh yeah, but the ones that imprinting is not seen in Costa Rica, there is no variation. So one idea is that maybe learning is there. I mean, learning is important in many contexts, but maybe that trait, the divergence in coloration, has not arisen in Costa Rica. So maybe that's why it helped it happening in the Panama clade, and it didn't really disappear in the polymorphic populations. Again, like learning is just important as a trait. That could explain why Costa Rica is not seen in Costa Rica, but it wouldn't explain why the polymorphic ones has not necessarily disappeared. In the polymorphic ones in the field, it's also hard to say because they could be learning from their social environment in the adulthood, but because it's polymorphic, there is probably a lot of gene flow, so it may as well that their mom is a different color than their own, so they may be imprinting on a different color. So that's why the disappearance of female mate choice and male aggressive biases, we cannot disentangle if it was another mom or they continue to learn. That was the idea of bringing them to Pittsburgh and raise them and see which developmental time is more important. But so far we cannot tell what you're asking. Yeah.
Yes, talk. I was wondering from an ecological perspective, how much if you were to vary something like competition, would that erode some of the behavioral choices, the biases, if there's more competition versus less?
You mean competition in the sense of like for resources or? Yeah, so one idea, I mean, these are different colorations and stuff, and color is usually tied to behavior, either for anti-predator behavior or stuff, but also to aggression, for example. So some preliminary results that suggest that the red phenotype is more aggressive than the rest of the phenotypes. So the red ones may as well be more good competitors than other phenotypes. This is something that we want to explore. Also, like frequency-dependent processes, which is more common, because if they bias their aggression and one phenotype is less common than the other, this will have an advantage because everybody kind of ignores them and they don't compete with them. So I would say that is a question of like how behavior relates to their competitive abilities, frequency-dependent processes. Yeah, and in general, they all fight for territories. The males that do not have a territory do not have success reproducing. That's probably the main trait that predicts reproductive success, have a territory. So they should not all be competing at some point, but yeah.
No, I thought it was interesting because if density increases.
Yeah.
in the population, perhaps it makes more sense to be more biased.
Yeah, exactly. Yeah.
Good talk. I was curious if any other traits, behavioral traits, social traits have changed, like the calls of the males across the islands. Like, do they have a bit of a different call? What happens if you,
Yeah, they do. They also change in size, which affect their calls. I try to not ignore, but I try to control the effect of the calls. In my playback experiments, I used one call that was the average of all of the populations. So more than taking it into account, I control for it, because otherwise my experiments would get huge for my PhD, like different combination of calls and colors, too much. Yeah, just to say that I have not tested that, but it's definitely an option. I control for it. They should all be listening to the same thing. It's likely that if I start to combine color and colors, and I mean, I was talking to someone about like the chemicals, because they do smell, these frogs, and in the water, the tadpoles smell their mom. So yeah, other modalities of sensory ecology are interesting. It can get huge, another PhD for someone else. Yeah.
Just preempted my question, maybe, but the chemical communication is interesting. I wonder if you put them in the dark, what do they actually show preferences to?
Yeah, great question. So yeah, so there is multiple pieces of evidence. I'm going to start with the tadpoles. So the tadpole imprinting story, it was nice, and they found good evidence that it was based on color. Another lab did some visual experiments of the tadpoles to ask how well are these tadpoles seeing, because in the water it's not like clean water, it's like water inside of a plant and it's kind of blurry. The results is that the tadpoles are blind, essentially. They couldn't find evidence that they are seeing color. So that was one way to, I don't know, to ask what happened with the experiment, the original experiment. And a follow-up experiment of the tadpoles is that they had the tadpoles in clean water now, but they purified like the chemical composition of the skin of the mom, and they put that in the water, and they looked at the neurons of the tadpoles, and the dopamine neurons started to trigger the begging behavior. But the tadpoles, instead of begging like this very like a specific behavior, they started to go around like this, like without a direction. And whenever they put a plastic frog in the water, that triggered the begging behavior. So at least for the tadpoles, the chemical gives the motivation and the color gives the direction. How do those two connect later in life, or how imprinting is occurring between the two of them, is kind of the question. And if they see in the dark, I don't think so. So I had a problem working with Panama and Costa Rica, because there is no variation in Costa Rica, there is variation in Panama. So for my experiments, I had a female and males of two colors. But for Costa Rica, because I cannot move frogs between countries because of legal reasons, I didn't have variation in Costa Rica. Didn't have a blue frog, for example. So what I did, based on our experiments, was to reduce the brightness that arrives to the frog with these filters in the dome. So the frog would look like brown, essentially, instead of red, and it did the job. So they saw the frog not as red, but as brown or something. Color couldn't essentially reflect towards the eye of the female. So yes, and some environments on the forest, sometimes you go there, it's really dark, and a red frog is barely visible. So it's very complicated to think about lighting. I think it's important to take it into account, but it complicates things a lot, and I don't think this is well in low conditions of light.
I think it was for the monomorphic populations, and I think it was male aggressiveness. I think in the tree, you showed that the ancestor was yellowish. There's
Yeah,
kind of a baseline signal of that. But I think there was one group where it looked like it kind of went green, suggesting that there's less of a signal for these dynamics. And I'm just curious what's going on there, if you have an idea.
Yeah, I mean, that's a good question. Oops. Well, I'm just going to say, oh, there is a question here, actually.
Oh, that was you.
Yeah, I just wanted to show you the map of Costa Rica and Panama. Okay.
Sorry, everyone. Back up.
Okay, yeah, I'm not going to move this. I wanted to show you the map of Panama and Costa Rica, but essentially the archipelago of Panama, of Bocas del Toro in Panama, is at the border of Costa Rica. So that population that you are talking about is just in Costa Rica. It's almost like a legal thing that we call it Costa Rica. It's just like noted there, and that was the one that was very yellow. So that show a lot of high proportion of attacks, probably because it's just so close to Panama. The other population of Costa Rica was in the border of Nicaragua. So it might be as well just a geographic kind of distance. It might as well be receiving some kind of influence from the archipelago of Bocas del Toro. But yeah, it was true for males and not for females. Yeah.
On average, how many tadpoles does each female have to care for at each time?
One.
Oh, just one.
Yeah, it's super specific because the tadpoles are obligate egg feeders. We have tried to take care of the tadpoles with fish food. Sometimes we give them the eggs myself, like I get it from the terrarium and feed them. They need their mom. So they are obligate. So the mom stops mating for like 30 days to take care of the tadpole. Yeah, it's very
So that's
specific.
a lot of energy to put into
Yeah.
one's offspring.
Yeah, and maybe that's why there is female choice, because I tested for male choice too for different females. Didn't find an effect because males increase their reproductive success by mating more. So they just mate indiscriminately. Females do have a choice because it's like more cost for them. Yeah.
Biological, and physical state kind of feeded on the different, sex preferences for interaction. How did you do ancestral state construction for this?
How did I do it?
Yeah.
So yeah, this one is a specific type because the usual ones are for discrete traits. Have you seen like the balls that have like half color of half color? This is a continuous trait from 0 to 1. So essentially you map the behavior on each population, which is a continuous value from 0 to 1. And then you have it takes into account the information of the branch lengths of the tree. And then there is a package in ARC by Liam Revel called PhyTools. You essentially have the information of the tree, the phylogenetic information. You have the branch lengths, like how deeply separate are the different branches, and then you just map the behavior. So essentially the color that you see is the likelihood that that trait has that particular value in that node. So if you were to have like a tree with two like terminals, and this one is 0 and this one is 1, this would be 0.5. It's just the likelihood that it is. And it's kind of like that with idea with more, it just goes from the top to behind. But you need that information, the relationships, branch lengths, and a trait that is continuous or discrete. You can also use it with something, other traits.
There are no more questions from the general audience. It's all good, Marco. Big round of applause.